end makes some sort of interactions as part of the acceptor step. The 5' end would be labeled there as position 1, and sort of that general area of the tRNA is recognized by the synthetase, but there's nothing special about the exact 5' end that's any better than the general fit of the acceptor step into the synthetase molecule. Does that work? I haven't thrown any non-chocolate, so I'm going to see how these fly. This might be sort of shotgun-like, but we're going to see what happens. Does that work? This is going to be ugly. Oh yeah, that's what all right, I've got heavier stuff. I'll go with heavier stuff next. Don't worry. Good enough. You might have gotten one of them. If you didn't, it's tough. So are we happy, right? So we've got to edit. We've got to deal with things. But basically, at this point, if you back up into what Eric Ross taught you and what I've been teaching you so far, is that there's some sort of regulation that ultimately allows RNA polymerase to initiate a promoter to generate a transcript. That transcript is then heavily processed. A 5' cap is put on it. Introns are spliced out. It's 3' polyadenylated. It's eventually exported from the nucleus. It's circularized. It's got exon junction complexes on it. It's getting ready, and we haven't even talked about the mechanism to actually load it into the ribosome. But once it's loaded into the ribosome now, we finally have molecules that are able to translate that RNA into an amino acid code and actually build polypeptides. And remember that you're making 100 trillion molecules of an individual protein per second in each of your bodies. Just keep that number going. But we have to think about what happens when our DNA is damaged and what ultimately happens to the genetic code and what are the effects of individual mutations. Because oftentimes in the laboratory, we use these individual mutations to tell us a lot about the function and mechanics and mechanisms of what could be happening. And so this is a line, that second line on this slide, is directly from your textbook. And it's utterly wrong, but it's in your textbook. But I want to point out why it's utterly wrong and how you should think about science. Evolution is about organisms staying alive or dying. The vast majority of organisms that have ever been on this planet are now dead. Right? Panda bears will be one day too, you will be one day too, it all happens, right? So the genetic code did not evolve to minimize errors. The genetic code evolved, and whatever organisms used that genetic code most efficiently survived and passed on their genetic information to the next system. So even these great big textbooks that you pay a lot of money for have dumb statements. But if we imagine individual mutations happening, and we think they're in the coding region of a gene, there's many different consequences of them, and they're important for how we do science in the laboratory. Oftentimes, if you change the last nucleotide of a codon, it has absolutely no effect on what amino acid is put in. CU followed by any residue will always code for leucine. And you can look that up on the table, and that's why whole blocks of them are dealt with by one individual amino acid. If you change the first amino acid in a codon, generally speaking, but not always, you're going to end up with an amino acid that is of similar size, hydrophobicity, and charge, if it has a charge at all. So you end up with very chemically similar amino acids. So having a mutation in the third codon is generally least problematic. Having a mutation in the first position of the codon is generally second most harmful. And having a mutation at the second position is really the whammy spot. It's really the one that changes things dramatically. You often go way off target in terms of what sort of amino acid would be put in place. And all of these sort of mutations have specific names that we're going to get to. And yes, they come up on your exam because they're critical to the way we do science in the lab. We often use silent mutations, which will be something we do in a minute, or missense or nonsense mutations, or frame system mutations. All have different consequences for how often the mutation will revert and how long the organism that's under study will be of interest to us in the laboratory. So these are the basic ideas of what can happen. If we consider the first mutation, let's imagine we go from a CUU codon to a CUC codon. Yes, the DNA has changed, the resultant RNA has changed. If that RNA is translated, the protein is exactly the same. So although it was a mutation, from the protein's perspective, it was a silent mutation. Very easy to understand the term silent, not a big deal. The second two always mess people up, and so I encourage you strongly to bang it into your brain however necessary, the difference between a missense and a nonsense mutation. If you change a codon at the first position in the example here, but you could do it in many different instances, from AUA to UUA, that is a single nucleotide change, and it will change the coding potential of that codon from isoleucine to leucine. So a missense mutation is exactly that. The codon is modified such that a different amino acid than the true genetically encoded one in the wild-type organism is now inserted. So if your protein was 65 amino acids long, it's still 65 amino acids long. 64 of the amino acids are exactly where they're supposed to be, talking to their nearest neighbors as they're supposed to be, and there's one new kid in the mix and one kid left. Basic idea. There is actually a really funny video that you can watch if you're ever interested of kids at Stanford actually acting out the steps of translation with flour bags exploding to be ATP synthesis and then moving around to be codons and anticodons, and they actually demonstrate how to missense mutation, where they built a big chain of people and they were all wearing blue shirts, and then they did it again and there was one red-shirted person in the middle. And it's really crazy. Yes?
So if missense mutation is you insert a different amino acid and it doesn't affect the function of the protein?
It could or it could not affect the function of the protein. Many residues in a protein, like if we look at the synthetase, that blue blob that was there, right? Some of the residues of that blue blob actually interact with the tRNA, and changing those residues would probably be very detrimental to the activity and accuracy of this protein. But changing other residues may not have much effect at all. But you can never tell exactly what will happen. The only definition of a missense mutation is that it's a mutation that leads to an alternative amino acid being put in place. It doesn't necessarily reflect that that amino acid has changed the activity of the protein at all. The last mutation is a nonsense mutation. Remember that the genetic code has 64 positions, that there's redundancy in it, that there's no ambiguity in it, and that there's 4 punctuation marks, a start site and 3 stop sites. Changing something so that another start site is there generally doesn't change anything. You just put methionine in it. It's a missense mutation. But if you change a codon such that it no longer codes for an amino acid, but rather functions as a stop codon, in our hypothetical example of a 65 amino acid polypeptide, if we all of a sudden have a nonsense mutation in the middle of it, we'll incorporate, say, 32 amino acids, and then we're instead of incorporating the 33rd, and the 34th, and the 35th, and so on, maybe the 33rd position has been changed so that the codon is now reflecting a stop codon, and thus nothing will happen there. Protein synthesis will end because the ribosome thinks it's reached the natural end, and once it's reached that end, it will release the polypeptide. Depending on where that stop codon is, where that nonsense mutation is, the polypeptide might be a quarter as long as it's supposed to, half as long as it's supposed to, one residue short of where it's supposed to. Sometimes those partial proteins have partial activity or altered activity or no activity. Sometimes they have the exact same activity as the full-length polypeptide. So again, just like the missense mutations, nonsense mutations are more classically detrimental, but not always detrimental.